The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
However, it is possible for DNA polymerase III to incorporate a non-complementary base to the new strand. This is known as a mismatch. A mismatch could result in a faulty protein being coded for, so it needs to be repaired. The first step in the mismatch repair pathway in E. coli is recognition of the mismatch by the mut-S homodimer. Mut-S recognizes the mismatch and binds to it. Then, the mut-L homodimer binds to mut-S, ready for a later step in the mismatch repair pathway. Meanwhile, up to a kilobase away, the mut-H protein recognizes a GATC site. Mut-H is a weak endonuclease, which will create a backbone incision between the guanine and adenine residues on the newly synthesized strand when activated. Mut-H is able to differentiate between the newly synthesized strand and the template strand by the lack of methylation markers on the newly synthesized strand. This is why the pathway is also known as methyl-directed mismatch repair. In order for mut-H to become activated, it needs to bind with mut-L. This causes the DNA to bend. The newly formed backbone incision allows an exonuclease to enter and begin excising nucleotides from the newly synthesized strand. The exonuclease excises the mismatched nucleotide and stops excising just beyond it. Now, DNA polymerase III can begin to resynthesize the new strand, inserting the correct complementary base where the mismatch used to be. The final step is for DNA ligase to ligate the backbone nick. The mismatch pathway is now completed, and all the newly synthesized DNA is complementary to the template strand.
The nucleotide excision repair process uses a set of proteins called UVRA, UVRB, UVRC, and UVRD. UVR stands for ultraviolet light repair of pyrimidine dimers. But remember that this system is also used to repair other types of DNA damage. In addition, DNA polymerase and DNA ligase are used to complete the repair. The process begins with the loading of a protein trimer onto the DNA double helix. This trimer consists of 2 UVRA molecules and 1 UVRB molecule. This trimer moves along the DNA, scanning for damage that distorts the double helix. When the trimer encounters DNA damage such as a thymine dimer, it stops. The UVRB protein remains at the damaged site, while the 2 UVRA proteins are released. UVRC then binds to the site and makes 2 cuts on the damaged strand. Both cuts are several bases away from the damaged site. UVRD, a helicase, then binds to the site and separates the segment of damaged strand from the rest of the DNA molecule. UVRB, C, and D are then all released from the damaged region. This leaves a gap that is filled with the proper nucleotides by DNA polymerase and then sealed by DNA ligase. The end result is that the damaged DNA has been replaced with the correct bases.
By turning genes on or off, epigenetics determine which genes are expressed, and these expression patterns are maintained through cell division. When cells of discrete tissues in the mammalian body divide, it is epigenetics that maintains their tissue identity. For instance, when your skin cells divide, they remain skin cells. Tissue state is controlled by the on or off status of sets of genes, thereby ensuring that when a skin cell divides, it does not turn into a hair cell or a bone cell. Most tissues in the human body contain exactly the same genetic blueprint. Tissue identity depends on which genes are turned on and off. While dozens of different histone tail modifications are possible, in this animation we will focus on just 2 important examples, acetylation and methylation. Acetylation of amino acids on histones generally correlates to gene activation. Acetylation reduces the positive charge of histone tails and repels them from the negatively charged DNA backbone in the packed nucleosome. This modification causes a slightly more open chromatin structure. Lysine and arginine amino acid residues on histone tails can also be methylated. Methylation is more complex than acetylation. Its correlation to gene activity depends on the context of where the mark is placed. For instance, when methylation marks are placed on lysine 4 of the tail of histone H3, usually this correlates with actively expressed genes. However, when methylation marks are placed on lysine 9 or lysine 27 of the tail of histone H3, genes are usually repressed. Epigenetic regulation is established and maintained by 3 major classes of proteins, or enzymes, that play distinct but intertwined roles during this process. Readers bind either active or repressive marks on chromatin. Writers place either active or repressive marks onto chromatin. Erasers remove marks from chromatin. These enzymes perform their job simultaneously, meaning that an active mark might be erased at the same time that a repressive mark is placed, and vice versa. How these enzymes work together is an active area of research. What is known is that epigenetic processes have the ability to turn on and off genes. The regulatory process is dynamic and reversible. That is, repressive marks can be removed, active marks can be added, and RNA production can resume, thereby converting repressed chromatin into active chromatin. The flexible nature of this process is critical for many aspects of early human development and life. The main thing to remember is that specific marks on the histone tails play a role in turning a gene on or off. On means that RNA is produced. Off means that no RNA is made. These marks help hold tissue identity and can be maintained through cell division.
Introducing the cell membrane. At just 7 nanometers wide, this phospholipid bilayer is covered in hundreds of thousands of different receptor proteins. These receptor proteins are each specialized to interpret different signals from the environment and then relay that information back into the cell. When a chemical messenger like a hormone or a neurotransmitter binds to a receptor protein, like a key going into a lock, the receptor protein activates corresponding effector proteins inside of the cell, initiating a cascade of chemical reactions within the cell. These are called IMPs, or integral membrane proteins. This receptor-effector protein relationship in the cell membrane is how a cell perceives and reacts to its environment. A molecule in the environment binds to a receptor protein, an effector protein gets activated, and the chemical release inside the cell makes its way back to our cell's nucleus, where the DNA is opened up, so a new protein can be made in the process of transcription. All the functions of DNA depend on interactions with proteins. For example, within the chromosomes, proteins such as histones compact and organize DNA. These compact structures guide the interactions between DNA and other proteins, helping control which parts of the DNA are transcribed. For a gene to be expressed, a sigma factor protein needs to bind to RNA polymerase for it to split the DNA and be able to access the blueprint. Different sigma factors are utilized under different environmental conditions. These specialized sigma factors bind the promoters of genes appropriate to the environmental conditions, increasing the transcription of those genes. The genes chosen to be activated and synthesized into new proteins are the direct result of signals from the environment interacting with receptor proteins in the cell membrane. This is the molecular basis for the environment being the primary factor in biological processes. Understanding this complex interaction between environmental conditions and gene expression leads to a marvelous new understanding of biological development.